Hello, I'm Vanessa Silva, and this is Francisco Bang Bang. Today, we will be talking to you about Zara Ebola virus and hope to educate you about this type of virus.
Ebola is known as a viral hemorrhagic fever. Ebola virus is a single-strand ribonucleic acid virus that occurs on the African continent which usually affects humans and non-human primates, such as monkeys. It is known to have a high mortality rate which is approximately 90% and can damage our immune system and organs which can eventually lead to internal bleeding. It is about 80 nanometers in diameter and about 970 nanometers long. It has a tubular shape that consists of three layers which are the glycoprotein surface, lipid membrane envelope, and an internal tubular helical nucleocapsid. The first layer of the virus consists of globular spikes that are formed from glycoprotein. It is usually 7 to 10 nanometers long. It would help with the entry of the virus into targeted their host. It could also be used as a mediator and can also help with binding and cell membrane fusion. The second layer is the lipid membrane envelope. It usually surrounds the third layer, which is the internal helical nucleocapsid, which acts almost as a shield. Now the third layer just helps with its process of a cycle when it reaches replication phase. Ebola belongs to the genus Ebola virus from the family Filavridae. It was once thought the Ebola virus is like a Marburg virus since they showed similar symptoms, but Ebola has 5 types of strains. The genome of Ebola consists of 7 genes that encode for nuclear protein, glycoprotein, virion protein 24, VP30, VP35, VP40, and a dependent RNA polymerase. It is usually transcribed the individual gene in their 3 to 5 order using the host ribosomes, tRNA, and mRNA into individual viral proteins. For them to replicate, they need enough nucleoproteins present to encapsulate neosynthesis antigenomes and genomes. There are about 5 types of species of Ebola, which are the Zaire Ebola virus, Sudan Ebola virus, Typhorus Ebola virus, Renston Ebola virus, and the Bundibugyo Ebola virus. The EBOV, Sudan, Typhorus, and Bundibugyo viruses are known to cause diseases towards humans rather than the Rensen virus. The Rensen virus is usually known to cause disease towards non-human primates and pigs, but not towards humans. There is another type of strain of Ebola virus which is called Bombali virus, but it's richly found in bats, and there is no scientific test that there may be a cause towards animals and humans. So the history behind Ebola is that it usually occurs in rural settings of West Africa. So each species of Ebola virus is named after the location where they originated. Yambalku is a village located in the country of Democratic Republic Congo, which is known today as Zaire. Ebola River translates to Black Death in the language of Lingala. Also, the year of 2000, since there was a confusion with the Marburg virus, it was changed to Zaire Ebola virus. The frequency of occurrence of the disease. So the Ebola virus is an emergency infectious disease that frequently occurs in Central, Eastern, and Western Africa. The first Ebola virus outbreak is the Zaire Ebola virus, which occurred in the Democratic Republic of Congo during 1976. During that outbreak, it had affected 318 villagers and killed about 280 of them. There were about 38 confirmed survivors during 1986 from Ebola. Those that have survived develop antibodies from this disease. The majority of survivors have long-term symptoms such as having joint problems and vision problems, and may experience fatigue with headaches and abdominal pain. As you can see on the right, this was a map during 1976 to 2018 or 2019. The brown dots is the Zaire Ebola virus, and then the blue is the Typhorus, and the yellow is the Sudan. And then the picture on the left is 2 nurses that were from Nagiama Hospital in Kinshasa, Zaire. They were known to be treating the third case of Ebola in that village. This chart on the right is the confirmed cases of Ebola that had occurred in Africa from 1976 to 2019. As you can see in the species category, the most common one is the Zaire Ebola virus, which is technically the deadliest strain throughout the other four. Throughout the 54 countries in Africa, the one that was hit the most during the outbreak was Guinea, Sierra Leone, and Liberia, which was the outbreak from 2014 to 2016. There were about 28,600 confirmed cases and about 11,310 deaths from the three countries alone.
Knowing about all those deaths, let me tell you how they were diagnosed. Most of the symptoms start to appear after 2 to 21 days. When Ebola was first starting to spread, many of the people diagnosing them thought they were dealing with dengue. Dengue has most of the symptoms Ebola has, such as headache, fever, muscle ache and pain, nausea and vomiting, and low counts of platelets and white blood cells. However, the main differences are that Ebola also includes diarrhea, bleeding, and low hemocritic and hemoglobin. The oral differences are different because dengue has a dry and crusty look and Ebola has a bloody look. The severity of the symptoms increases throughout the duration of the disease. There are four phases to Ebola. The first phase is called the early febrile and it consists of the first 3 days. During these days, the most common symptom is fever, but it may also be accompanied by malaise, fatigue, and body ache. The second phase is called gastrointestinal and it consists of day 3 to 10. The most common symptoms are epigastric pain, nausea, vomiting, and diarrhea. It may also be accompanied by a persistent fever, headache, conjunctival injection, abdominal and chest pain, arthralgia, myalgia, hiccups, and delirium. From day 7 to 12 is the shock or recovery phase. During this phase is when things can take a turn for either better or worse. If a person goes into shock, they will have diminished consciousness or be in a coma. They may also experience a rapid thread pulse, oliguria, anuria, and tachypnea. If a person takes a turn for the best, they will experience the resolution of gastrointestinal symptoms, increased appetite, and increased energy. The turn for the worst will lead us to our final phase called late complications. After day 10, this is when a person would experience gastrointestinal hemorrhaging. This can lead to a secondary infection such as oral or esophageal candiditis and persistent neurocognitive abnormalities. The pathogenicity of Ebola is broken down into three phases. The first phase starts with animal reservoirs that aren't humans, such as bats. The virus then enters the human body through a break in the epithelial continuity, skin abrasions, or mucosal lacerations. Once inside the body, it spreads to the lymph nodes, liver, and adrenal glands. This leads us to phase 2, where there is a viral multiplication in the cells. From here, it has three options. One of the options is when it attacks the dendritic cells, it leads to the activation of T cells. This leads to premature contraction due to apoptosis. Phase 3 of this route will lead to impaired immune response. The second option would be attacking the neutrophils. This would lead to activation and degranulation, which would trigger the systemic inflammatory response. Phase 3 of this route would end with fever, malaise, and gastrointestinal symptoms and possible sepsis. The last possible outcome is when it attacks the macrophages, which leads to endothelial leakage, and when it attacks the monocytes, which leads to multiplication in liver cells. The final stage here leads to a hemorrhage. There are a variety of ways in which Ebola can spread. The most common way that the virus was spread was through bodily fluids. In 2014, there was an outbreak in Guinea, and 68% of the cases were spread through bodily fluids. They have a tradition where they pass the deceased body down into its grave. Touching the diseased bodies helps spread Ebola to more than half of the Guinea population. Droplets such as stool, semen, saliva, and breast milk are also contributors to the spread. Also included in droplets is accidental infections from non-human primates. In the 2008 Ebola case from the Philippines, the virus was traced back to a pig that wasn't cooked all the way through. In addition, personal protective equipment, PPE for short, such as gloves, masks, gowns, and skin, etc., may break and allow the virus to enter. Improperly disposing of sharp instruments is also an easy way for viruses to spread. This was the reason for the outbreak in 1976 in Sudan and Zaire. The infection was spread by contaminated needles and syringes. Improper hygiene and sterilization spread Ebola like wildfire. This was the worst case in the history for an 88% mortality rate. When this virus was first seen in the 1970s, there really wasn't anything that the doctors and nurses could do to help treat the disease. It was a fairly new thing, so they didn't really understand it. The only thing they could do was to provide fluids and electrolytes, use oxygen therapy, and use medication to help with dry and wet symptoms. For a long time, the thought of a vaccine to treat Ebola sounded impossible. For decades, scientists tried to find something to treat this horrific disease. Finally, in December 19 of 2019, a vaccine was finally approved by the FDA. The name of this vaccine is Iverbo, and it is given to individuals who are 18 or older. This vaccine was made possible thanks to the study conducted in Guinea during the 2014 and 2016 outbreak. They were able to successfully genetically engineer a vaccine that contains a protein from the Zaire Ebola virus. There was a study that was conducted by Dr. Fisher and another scientist named Mr. Judson from the Virus Ecology Unit at Rocky Mountain Laboratories in Hamilton, Montana. They wanted to know how long Ebola could live on a surface, so they tested Ebola on 3 materials found in an Ebola treatment unit in West Africa. They tested utility-grade stainless steel washers, plastic, and Tyvek, which is similar to PPE suits. They tested it in an African tropical condition of 27 degrees Celsius and about 80% relative humidity. However, they also conducted the experiment in a stimulated climate-controlled hospital condition of 21 degrees Celsius and 40% relative humidity. In the tropical condition, the virus is only able to live on the stainless steel and plastic for about 4 days, and it lived in the Tyvek for about 5 days. In the stimulated hospital conditions, the virus was able to live on the stainless steel for about 7 days, it lives on the plastic for about 11 days, and it lived on the Tyvek for about 15 days. Luckily, the tropical conditions in Africa reduced the amount of time the virus can live on a surface.
The topic that we have been focusing throughout the research was Ebola's mutation. The Scholar article is from a Berkeley library and the popular article is from the CDC. Both articles state that the viral family Filariidae is an old virus which is only transmitted by bodily fluid. It also states that the Ebola virus won't mutate into an airborne virus due to how usually the stable mutation is. However, it seems that Berkeley article is in depth with information about Ebola, stating how high the mutation rate and rapid rate of replication, which allows it to evolve quickly. They also stated that due to having a high mutation, we should be worried because as it evolves, it can render standard testing, which becomes inaccurate and challenging to diagnose this type of disease in an infected person.